I-lens laminae. So they grow, this is a lamina, or a lens that's been cut, and you can see the different laminae. We call this the nucleus. And you can just take these sections off and run them through the IRMS and get your isotope numbers. They separate very easily. The reason that they're conservative is because the cells that grow, that accrete onto the outside, are made of an optical protein that's very durable called crystalline. And once those cells are made, to make it even clearer, they remove all of the organelles. So the nucleus and mitochondria, all of the organelles are taken out of the cell. So there's no way that the cell can make any more protein. It doesn't have the apparatus for it. So that locks in the carbon and nitrogen in the crystalline. The same thing's going on with all vertebrates. We haven't checked very many of them, but in theory it applies to all vertebrates, and it also applies to squids and octopi. This is some of Amy's work that was published in PLOS One last year. And this is just an example of her comparing the left and right eyes for agreement from an individual fish. So this is early in life, and this is capture. And you can see that there's very good agreement and that the trends are interesting. So we've then taken this method and started to apply it more broadly. This is Jenny Ostroff's data from a manuscript that's about to probably go out next week. This is again tile fish. This fish here on the right. And this is its entire life, early life to the point of capture. Notice these are radial midpoints. Our units are millimeters. They're not age yet. They're not time. We're still dealing with just millimeters. And you can see that the carbon and nitrogen follow the same trajectory. They're very strongly correlated. Given that the isoscapes are orthogonal, you can't get this pattern if the fish moves. You can only get that pattern if it stays put. And the reason that it increases is because its trophic level is changing as it grows larger. We repeated that, and this is 36 different individuals, and they all had very high, this isn't even the highest correlation, this one is 0.96 for an R-squared value. But they're all very high. You can see some deviations which we think are actually the prey coming in from other areas rather than the fish moving. And the reason that these fish just show what's called an ontogenetic trophic growth rate, they show that basically their trophic level increases as they get bigger without anything else going on, is because they live in burrows. And they dig the burrow when they're small and make it larger. It's a lifetime project. And they're thought to stay in the same burrow their entire lives. There are some observations of them switching burrows, but they form communities called pueblos. And they don't leave, they're not thought to leave their pueblo. So that's an example of fish that from our data, our I-lens isotopes, we don't think they move. And then, of course, you read the literature and everybody says, no, they don't move. They stay in there. They're dedicated to those burrows they spent so much time digging. And they're eating the fish and crustaceans that go near the opening, but they're protected from large predators in the meantime. You compare that with a gag, which is a type of grouper, and you can see trophic growth indicated in the nitrogen, but the carbon shows estuarine juvenile development, then movement to the clear, shallow coastal waters, polyhaline seagrasses, where you have a lot more benthic algae contributing to the biomass of the fish. Ultimately, it's not directly. They don't eat the algae. They eat the things that eat the things that eat the algae. So, but you see that, and then they migrate offshore, which is also phytoplankton-based like the estuary. And so you get this really interesting story. And this is actually a low-tech method. Anybody could separate those laminae and mail them off and get data from them. This fish is one that moves. We knew it moved, And we have actually a lot of different species. We've done all these methods. I'm just showing you examples, but we've repeated these methods on a lot of different specimens and species. So what we have planned for the future, extend our isoscapes to the entire Gulf of Mexico. We're hoping to go to Mexico next month on the Weatherbird, take the Weatherbird all the way across the Gulf and work the Mexican coastline. We have extended the method to sharks. We have the data, I believe, Craig? Yes?
Yep, some of the data. Yep.
Some of it. And we've started, Brenna Meath is another student in the lab who we've done test dissections of squid eye lenses, and it works fine. Combining the eye lens data with otolith microchemistry is exotic. No one else is doing that. Jen Graneman will be doing that soon. We're also combining it with PAH data from the Morosky lab. And Amy is busy. Also, she has a lot of data. We're trying to convert that millimeter axis into actual time by comparisons with otoliths. Greg and Amy are busy separating the trophic and geographic effects in the eye lens data by isolating suites of amino acids from eye lens layers and then measuring the isotopes within those different amino acids. That's an exotic method by itself, and then when you apply it to eye lens laminae, no one else is doing anything like it. Ultimately, we're being very careful in building this in a stepwise fashion, but ultimately the goal is to have maps of the individual fish movements. And that's, again, where a lot of students are going to be doing that, including students from the Stallings lab. We've been focusing on the oil spill because that's what pays us. And we have a number of students that want to start adapting these methods to fisheries-related questions rather than oil questions. And finally, the best way to confirm all of this is with a captive study. So we have that scheduled and funded down at Moat. Jen Graneman will be working on that. So by pioneering these new methods and combining them with existing methods, we've basically developed a truly unprecedented forensic capability. And with that, I'll take any questions.